The early morning, humid air of Florida feels thick. 6:32 a.m. and I find myself at Bayboro Harbor, home of USF College of Marine Sciences Ocean Circulation Lab. The atmosphere is quiet, seemingly because no one's caffeine has kicked in yet. That changes quickly. Myself and 13 others are embarking on a 5-day cruise led by Chief Scientist and Physical Oceanographer Jay Law aboard research vessel Weatherbird 2. With support from the Florida Institute of Oceanography, we are embarking on an interdisciplinary cruise under the College of Marine Sciences' newly established Collaborative Oceanographic Research and Education Program, abbreviated as CORE. The program aims to provide an interdisciplinary element to the existing buoy and mooring work done by the Ocean Circulation Lab every year, by incorporating other focus areas such as biology and chemistry. It is an opportunity to allow these students a step out of their specialization and see what it's like to work as a team with physicists, chemists, biologists, and even journalists. Not only is everyone learning from each other, but having a better understanding of how other disciplines operate makes one more well-rounded as an individual professionally. Waving goodbye to land, we set out into the Gulf of Mexico with 14 stops ahead of us. From even before setting sail, everyone was preparing for the 5 days ahead. Once on the boat, they kept working. Everywhere I looked, something was happening. Divers were checking air tanks for the upcoming dives. Individuals from the lab were prepping the vials, bottles, and equipment, and pockets of discussion going over protocols can be heard over the roar of the boat engine. The biggest piece of this expedition is the buoy and mooring work. If you've ever lived in the USA's coastal east, you've probably worried about a hurricane at some point. These buoys play a huge part in collecting that data from when storms are approaching. Air temperature, wind, humidity, water temperature, and air pressure sensors, just to name a few. These all help in everyday weather, but air pressure and water temperature may be the most notable for tracking hurricane strength and predicting storm intensity. I was able to briefly talk to Sam D'Angelo, a USF undergraduate with 6 buoy deployments under her belt. She is the student lead for the mooring component of the cruise.
We have 4 real-time in-situ buoys along the West Florida shelf. We have a MET package on the top, so we have air, pressure, speed, direction, humidity, like relative humidity, barometric pressure, so it's a whole bunch of stuff. It's really cool. Like during Hurricane Ian, you could see the pressure drop, you could see a spike in the ADCP data. It was super cool.
It's grueling work to deploy buoys. It's full physical labor with the Florida heat and sun blaring down on the team. Though, watching it all play out was akin to a choreographed dance number with how efficiently everyone works together. Board the buoy, rig the chain, release, repeat. When mooring work isn't going on, scientific diving training takes up the boat deck. Before the dive team goes under, the most important factor is safety. The handful of dives are to recover physics instruments and to survey fish populations at natural and artificial reefs. My first thought when I heard this was, man, I didn't know perfect memory and fish identification skills were a prerequisite to being a scientific diver. Well, as it turns out, a special whiteboard and charcoal pencil are used to write underwater. Cameras help documentation too. One dive is to retrieve a bottom-mount device that rests on the ocean floor and records data on water currents. This data gives researchers insights into water circulation, which drives nutrients throughout the ocean, effects that can drastically change the ocean environment all the way up the food chain to us. There is not much to see from the surface once the divers go under. However, when day turns to night, it is a much different story. Each diver wears a high-visibility glow stick that allows them to be seen from above, and the light scatters beneath the surface. Artificial neon light meets the natural darkness of the world in a beautiful visual that hypnotized me. For those who have seen divers go underwater at night, this probably was nothing to them. But to me, someone who's never been on a boat at night, it was such a cool sight. Pair that with the bioluminescence flickering in the water, and the clear skies where you can make out the Milky Way, It felt like another world. Once we started moving again, we even had some dolphin friends to accompany us. Waking up to mechanical movement sounds above, I find the deployment of the CTD rosette into the water. Over the course of the trip, we stop at 9 locations for data collection. Various stops stretch from just off the coast of St. Petersburg to over 100 miles offshore. The wide range of stops allows for a wider range of data in multiple environments, with a depth ranging from 15 meters to 750 meters, or roughly 50 feet to 2,460 feet, the latter equating to about 8 football fields. Going further out from shore and deeper into the ocean gives scientists insights on the differences between each location and raises further research questions when things differ from previous data sets. It's particularly important for the chemistry team that is collecting water samples. One location may show a high concentration of a certain type of algae, whereas another may show a complete lack of it. Around the rosette, you can see these cylinder containers called Niskin bottles. These are built to collect water at varying ocean depths. There's also a device that collects data on conductivity, temperature, and depth of the water called a CTD. All this data helps monitor ocean variables like water currents. As the team monitors the CTD data, they also trigger the bottles to close at certain intervals. In this case, near the surface, ocean floor, and the area of maximum chlorophyll in the water. Once the rosette is back aboard, the team works to collect the water samples and brings them back into the lab for processing.
This is the first time we're doing this specific transact.
That is Lily Verrill, an undergraduate at USF.
As we are going all the way to the end of the Florida Channel. Many cruises go out to like this distance, but we're going all the way out. A lot of people don't go out this far, so we don't have a whole bunch of samples this far. Because this is a nice, easy transect that the school that we're hoping to maintain. It'll be really interesting because we'll be getting data from areas that a lot of people don't regularly visit.
She oversees the chemical sampling. Each week she spends 3 to 5 days in the chemistry lab on campus. Her experience helps her guide fellow students through water sampling. These samples help scientists identify significant changes in the water and the greater ecosystem. One of the most prominent things they can find is traces of harmful algae blooms, such as red tide, which can kill marine animals and affect the respiratory system of humans. Whenever the rosette gets deployed, this tool, called a Bongo Net, collects microscopic organisms such as phytoplankton in the water at the same location. Professor of Biology at St. Petersburg College, Lene Baimi, leads this effort. To a max depth of 200 meters, 656 feet, the Bongo Net collects and filters seawater into 2 containers, which allows the team to bring the water samples back inside the lab to look at under a microscope. From here, Baimi can identify critters in the water. Specifically, her research interest is in zooplankton, which are the animals that eat phytoplankton, or, as she likes to call them, nature's little Cheez-Its. Some of the creatures bounce around when seen under the microscope, and others casually float within the water sample. Though they wouldn't be appealing snacks to us humans, these critters would be an appealing snack to the fish in our oceans. In a way, they are small but mighty. It's because their impact reaches far greater than their size and goes all the way up the food chain. With all these elements of oceanography coming together, it helps shape our understanding of the ocean. Changes in ocean conditions impact the little critters that live within it. Those impacts, in turn, influence larger marine species. In dire circumstances, animals can struggle to adapt or die if conditions change too drastically. Even land animals, yes, including us, depend on the health of the ocean for survival. It all interconnects. Across these 5 days, passion for science and the work these students were doing was always present, palpable even. Though many of the students had been on boats before, even the Weatherbird in the past, The sense of accomplishment and a greater love for the water was there. It is an attestament to why interdisciplinary activities are so vital to academia. I'm not a scientist of oceans, and statistically, you probably aren't either. But what we are is scientists of exploring the complex world we live in, observing, experimenting, and reasoning to the best of our ability. Finding what makes the world tick, what makes us tick, what our path will be and how it may change. It's easy to get caught up in the rush of the world around us, but sometimes it is good to slow down, to listen, to understand another story. There are so many interesting stories waiting for us in the world. So, take a chance. Do something you would have never thought you'd be able to do, because you will never know where you'll end up and what it will teach you.